Hey everyone, and welcome to the first video of our Bioinformatics with Python series. I'm super excited to get started. If you have ever been curious about how we are using languages like Python and R to analyze DNA, RNA proteins, you're in the right place. We are going with very basics in this series. So we will go from the absolute basics to analyzing real biological data step by step. So today we are starting with the most fundamental building block of all, the biological sequence. We will learn how to represent a DNA sequence in Python and perform some of the most common basic operations on it. For this, all you will need for the time being is some app that supports Jupyter Notebook like VS Code or any other development environment. And also you need to make sure that you have Python installed. For this course, I'm going to be using the Python version 3.13.7. All right, so let's jump right in. First of all, as you can see, I've got a new Python notebook opened in VS Code. At its core, a DNA sequence is just a string of letters A, T, C, and G. So in Python, the perfect way to represent this is with a very simple string. So I am assuming that you know the basics of Python for this course. If you want me to create a course for understanding basic Python before you get started with bioinformatics, please do tell me in the comments and I will try to create a course for that as well. So yeah, let's start. Let's create our first DNA sequence in a new cell. I have created a new cell and I will type this DNA seq. Let's create a simple sequence A T G A C G P T A G A C C. All right. So this with this we have created a simple sequence, and now I would write a code to print this. So when I run this, this is what we get: a simple DNA sequence. All right. So once we run it, here we have stored a DNA sequence in a variable called DNS-SEQ. Simple, right? So now that we have a sequence, what is the first thing that a biologist might want to know? So I think that would be how long is it and what is its composition. So Python makes it very simple to getting the length of any string. So with a function called len. So let's try it. Right? Sequence length is equal to len DNA sequence. Right. With this, we have the sequence of our DNA sequence assigned to this variable. Now we will print this. The length of the sequence is basis. All right, this should do it. And now you can see the Python has calculated the length of the string, and it is giving us, we have a total of 14 bases in our string, in our DNA sequence, right? So now what about the number of adenine, thymine, cytosine and guanine? These are the nucleotide composition, right? So we need to find that out as well. So Python strings have a very handy method called count. So we are going to use that only, right? So for that we will use this. A count is equal to DNA sequence dot count and whatever you want to count in that string. So let's say A. Now I will do the same for the other nucleotides T, then we have C and G, and we will change the letters here. We have T, C and G. What this will do is this will count the exact number of letters in that string, the nucleotide bases, and then it will assign that value to these variables. Now we will print is a count. Copy this and then P C G T C and G. Now when we run this code, yes, we can see here that it has counted the exact number of times that nucleotide base is appearing in this sequence. And there we have it. We can see exactly how many of each basis we have. A very common metric derived from this is the GC content, which is the percentage of the sequence that is either G or C, and it is often important in determining the stability of the DNA, as you might know. So let's calculate that. GC content is equal to G count and plus C count divided by sequence length, which we already calculated before, into 100. So this is the first simple formula of GC content, and now we will print this. The GC content is percent. Alright, now we will try to run this code and let's see what happens. So here we have it. The .2f in the print statement over here, it just formats our number to two decimal places. As you can see, calculating these fundamental properties is really straightforward. Alright, so I hope this is all with the basics of the initial video. We will move forward with the other things like transcriptions and rest in the next video. I hope you enjoyed this lesson. If you have any questions, do ask them in the comments. Thank you. See you in the next video.